I now call to order the lecture portion of the Society's 2527th meeting in what is now the 154th year since the Society was founded on March 13th, 1871. Good evening, everyone. My name is Larry Milstein. I am the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry to as large an audience as possible. Welcome to our members, guests, and friends to the lecture portion of tonight's PSW meeting and to the lecture by Davey Smith on the last gift, finding a cure for HIV. To start with, let's thank PSW's sponsors. Society is grateful to PSW member Mike Helton and Helton Associates, LLC for their full-year sponsorship. Thank you, Mike. And to PSW member Joe Schulman and the Schulman Foundation for their full-year sponsorships. Thank you, Joe. And the sponsor of tonight's lecture, PSW member Marlene Becky, in memory of Ivan Becky, a longtime PSW member who was Director of Advanced Space Flight Systems at NASA. Thank you, Marlene. The Society needs your support to meet its expenses. So if you've not given or feel you can give more than you have, please do not hesitate to donate. And now is a particularly auspicious time, not just because it's the giving season of the holidays, but because PSW is the beneficiary of a very generous matching grant. Every dollar you give will be matched 2 to 1, up to the first $5,000 of donations to PSW. The match runs through January 31st. So you can choose the most favorable of the 25 and 26 tax years. So please give. I'm pleased to announce the following new member. Steve Shoshinsky, Chief Systems Engineer at SAIC, interested in physics and aerospace science and engineering, who learned of PSW from searching the internet. Please join me in welcoming Steve to membership. Our speaker tonight, Davey Smith, is already a member, having previously spoken at PSW during the COVID years at the 2425th meeting on September 25th, 2020. That talk was given via Zoom. All members are entitled to a signed copy of Compendium Volume 17 of the Bulletin of the PSW. If you've not received yours and you're a new member, please come see me after the lecture. All members are invited to the pre-lecture receptions and dinners with the speakers for camaraderie and interesting discussion. If you're interested in membership, please join. You can access a membership application on the PSW website by clicking the blue Join button, believe it or not, on the homepage at the upper right-hand corner. It's very easy to see. If you're here in the Powell Auditorium, you can access the application using the QR code on the business cards on the tables at the back of the room. And if you have any questions, you can ask any of the people who are members of the board, including me. We'll be happy to tell you whatever it is you'd like to know about PSW and becoming a member. Everyone with a genuine interest in science is welcome to apply. So please join. Recording Secretary Scott Matthews will now present the minutes of the 2526th meeting and the lecture by Rosalie Lopez on volcanism and cryovolcanism throughout the solar system. Scott, the stage is yours.
Thank you, Larry. Good evening. On December 5th, 2025, members of the Society and guests joined the speaker for a reception and dinner at 5:45 p.m. in the members' dining room at the Cosmos Club. Thereafter, they joined other attendees in the Powell Auditorium for the lecture proceedings. In the Powell Auditorium of the Cosmos Club in Washington, D.C., President Larry Milstein called the lecture portion of the 2,526th meeting of the Society to order at 8 p.m. Eastern Time. He began by welcoming attendees, thanking sponsors for their support, announcing new members, and inviting guests to join the Society. Scott Matthews then read the minutes of the previous meeting, which included the lecture by Jeffrey Alexander titled The Real Returns on Public Investments. The minutes were approved as read. President Milstein then introduced the speaker for the evening, Rosalie Lopez of the Jet Propulsion Lab. Her lecture was titled Volcanism and Cryovolcanism Throughout the Solar System. The speaker began by saying that you can't understand volcanoes on other planets if you don't understand volcanoes on Earth. She said that the solid surface of planets represents the interplay of four major geological processes: volcanism, tectonism, impact, and erosion. Lopez showed a map of the Earth indicating that the vast majority of volcanoes occur on tectonic plate boundaries. She listed different types of volcanic activity and the types of landforms they produce. Icelandic and Hawaiian, forming fissures and shields. Strombolian, Palaean, and Vulcanian, forming cones with mild to moderate explosions, and Plinian and Ultraplinian, forming cones with large explosions. She said that the type of volcano depended largely on the type of rock, and that the shape of the volcano gives clues about the type of material involved. Lopez then discussed the surface of the moon, indicating that the large dark areas on the moon, called mare, are the result of lava flows, similar to Icelandic eruptions. She showed images acquired by the Apollo 15 mission, which include lava flows, lava channels, eruption cones, and domes. She showed images acquired by the Messenger mission, which indicated pervasive volcanism on the surface of Mercury. The speaker said that impact craters are almost always circular, while volcanic craters are formed by collapse and are therefore often irregular in shape. The speaker showed images from the surface of Venus, reconstructed from radar data, which indicate the presence of massive volcanoes, including lava flows, calderas, and, quote, pancake domes. She showed images from the Magellan mission, which support the conclusion that active volcanism on Venus has occurred as recently as the 1990s. The speaker showed topographic images of the surface of Mars, saying that Mars has the largest volcanoes in the solar system, including Olympus Mons, which is 26 kilometers high and about 600 kilometers in diameter. She discussed the possibility that an eruption of Olympus Mons could have melted Martian permafrost, creating flash floods and landslides, the remnants of which are visible in the topographic data. The speaker showed images from the Curiosity rover, which indicate the presence of, quote, clasts of elemental sulfur, consistent with volcanoes observed on Earth. The speaker then discussed cryovolcanism, essentially volcanism where the erupting material is not molten rock, but primarily water with other components. She showed images of a suspected cryovolcano on Ceres, a dwarf planet in the main asteroid belt, with a layer of liquid water under its crust. Lopez showed several images of Io, one of Jupiter's moons, claiming that it was the most volcanically active body in the solar system, with at least 174 active volcanoes and hundreds of inactive volcanic features. Some of these images showed volcanic plumes rising hundreds of kilometers above Io's surface. She described the gravitational process by which Jupiter's other moons would create tidal forces on Io, supplying the energy to keep Io's core molten and drive volcanic activity. Lopez showed thermal images of Io acquired from the Galileo mission using the Near-Infrared Mapping Spectrometer, or NIMS. She said that more recent analysis of NIMS data allowed her to confirm the presence of more than 300 active volcanoes on Io. Although the 2006 edition of the Guinness Book of World Records, which acknowledges her as the record holder, credits her with discovering a mere 71 active volcanoes. Lopez describes the types of volcanic activity observed on Io: Lokian, lava flows or flooded calderas; Promethean, long-duration lava flows; Pellinian, explosion-dominated events; and two other rarer types, which are possibly sulfur and silicon dioxide flows. She showed images acquired from the Juno mission in 2024, which clearly indicate the presence of, quote, hot rings associated with lava lakes, indicating that lava lakes were the most common form of volcanism on Io. For comparison, Lopez showed a number of images and videos of lava lakes on Earth, all of which she visited personally. The speaker discussed cryovolcanism on Jupiter's icy moon Europa. It is widely believed that Europa has liquid water oceans below its icy crust, which could potentially support extraterrestrial life. Lopez showed images of plumes ejected from the surface of Europa, presumably due to cryovolcanoes. She indicated that the Europa Clipper mission, which launched in 2024, could confirm this hypothesis when it reaches Europa in 2030. Lopez showed images of Enceladus, one of Saturn's icy moons, also believed to harbor a liquid water ocean. These images, acquired by the Cassini mission, show massive cracks in the icy surface and geyser-like jets of water vapor, molecular hydrogen, and other volatile compounds. The speaker showed possible evidence of cryovolcanism on Titan, another one of Saturn's moons. Radar data from Cassini indicate the presence of a crater about 1 kilometer deep in close proximity to a peak about 1 kilometer tall, structures that are commonly associated with volcanic activity. Showing images from the New Horizons mission, the speaker speculated about the presence of cryovolcanoes on Pluto. The speaker ended her talk by discussing the possibility of volcanic activity on exoplanets, saying that a sodium cloud, consistent with volcanic activity, has been observed near an exomoon orbiting WASP 49b. The lecture was followed by a question and answer session. A member asked Lopez how she became interested in planetary physics as a little girl or a young lady. Lopez responded that she grew up hearing about the Apollo missions and wanted to be an astronaut. But given her very poor eyesight, she thought her chances in the space program were slim, so she decided to study astronomy. She said that she was inspired by Frances Poppy Northcott, one of the few women working in mission control during the Apollo missions. A member asked if cryovolcanoes could exist in close proximity to magma volcanoes, and if so, could an eruption of one trigger the other? Lopez responded that although this situation has not been definitely observed, it is a real possibility, and something that features on Io some of the features on Io could be potentially explained by such proximity. A member on the live stream asked Lopez if she had any thoughts about what heat sources drive volcanism on Pluto. Lopez responded that the energy source was very likely tidal in nature. After the question and answer period, President Milstein thanked the speaker and presented her with a PSW rosette, a signed copy of the announcement of her talk, and a signed copy of Volume 17 of the PSW Bulletin. He then announced speakers of upcoming lectures and made a number of housekeeping announcements. He adjourned the 2,526 meeting of the Society at 9:43 p.m. Temperature in Washington, D.C., minus 2.8 degrees Celsius. Weather, cloudy. Audience in the Powell Auditorium, 71. Viewers on the live stream, 28, for a total of 99 viewers. Views of the video in the first 2 weeks, 314. Respectfully submitted, Scott Matthews, Recording Secretary.
We now turn to tonight's lecture by Davey Smith. Davey is Assistant Vice Chancellor for Clinical and Translational Research at the University of California, San Diego, where he is also Chair of Faculty in the Department of Medicine, Chief of the Division of Infectious Disease and Global Public Health, and Professor of Medicine. He's also co-director of the San Diego Center for AIDS Research and holds the Florence Seeley Ryford Chair in AIDS Research. Davey's primary research focuses on HIV transmission and reservoir dynamics, understanding how the HIV virus spreads and persists within the body, and on discovering new ways to interpret and understand those processes. He is a pioneer in viral phylogenetics and characterizing the drivers of superinfection. During the COVID pandemic, he focused on worldwide efforts to find safe and effective COVID treatments, serving as protocol co-chair for the ACTIV-2 treatment study and spearheading collaborations among the 17 NIH-funded Centers for AIDS Research. Davey is an author on more than 400 scientific publications, including seminal works on HIV transmission, viral reservoirs, and novel antiviral therapeutics. He is a recipient of many honors and awards. He earned a BS in biology at the University of Tennessee, Chattanooga, an MD from East Tennessee State University College of Medicine, and an MAS in clinical research at UC San Diego. All questions will be held until the question and answer period following the completion of the lecture. Davey, the stage is yours.
Thank you, Larry. Thanks, everybody. So today we're going to talk about the Last Gift program. And since I know there's the minutes are being taken so rigorously, I will make sure I don't say any bad words. I can't promise that. So we're going to talk about HIV cure. We're going to talk about viruses. We're going to talk about reservoir, and we're going to talk a lot about dying today. So the last time that I talked at this esteemed meeting was during COVID, when I was the director of one of the Operation Warp Speed groups trying to find outpatient therapeutics for COVID. But one of my biggest passions have been trying to find a cure for HIV. And to do that, we need to really look at microenvironments. So we're going to look at human tissues and what does HIV do in those, and that's the big takeaway for today. These are my disclosures, but the biggest disclosure is today I want to educate and I want to inspire. So we're going to start off with inspiration. So here's one of my favorite quotes. It's, "My, you chivalric fool, as if the way one fell down mattered." Well, when the fall is all there is, it matters. And this is a good quote, but really the inspiration of this project was a patient that I had when I was an intern. I learned how to do lumbar punctures on him pretty much every day because he had this infection in his brain called cryptococcal meningitis. And it was causing the swelling in his head. And if I didn't go in there and take the pressure off by taking a spinal tap every day, it would have popped his head. And that was how I learned how to do lumbar punctures. And that was in 1996. It was horrible. And but that was when the HIV medicines came out as a cocktail. And he was volunteering for every single one of those studies that he could get his hands on. And he was one of the first ones to get the cocktails. And he lived, and he became my patient for a very long time. And then he died. He got pancreatic cancer and was dying. And he really wanted to join research studies at that time of his life because he knew that it saved his life before, and he had always believed in research. But because he was dying, he was not allowed to participate in particular research that he wanted to. And I was thinking, well, that's kind of a bummer. So my question for you as the audience today is this one. Would you be willing to participate in a research study at the end of your life if you had a terminal illness and only 6 months to live? So think about it. It turns out that research ethics oftentimes consider people at the end of their life to be too vulnerable to make that decision for themselves to participate in research. And that was what my patient was encountering. So we asked this question to a whole bunch of people. We asked this question to people who had HIV and people who didn't have HIV. I want to introduce you to one of my colleagues, Sarah Gianella, who was a postdoc in my lab for a long time and then grew up to be a professor of her own right. And we worked very closely on the Last Gift together. But one of the studies we did was how many people would be willing to participate in research at the end of their life. And as you might guess, 80% of people said, yeah, I would love to do that. How many people would love to give organ donation? Well, about 76 to 80% of people also said, yeah, take my organs, I'm done with them. And then we asked them some very provocative questions. And this is the first set, and you'll have to wait till the end of the talk to get the other provocative questions. But the first ones were, would you be willing to give up any of your days, and you only have a few left? Would you be willing to give those up, some of those up, to participate in research? And it turned out that about a third of people would be willing to give up days in their short period of time just to be able to participate in research. And for people who had HIV, almost a third of them would be willing to give up a month of 6 months that they had left. So then we're like, okay, this might be something we want to pursue further. This is something that people want. So we performed we wanted to do studies in living people, but it's hard to reach the real reservoirs for HIV. If I want to cure HIV lives everywhere in somebody's body. It lives in their brain, it lives in their spleen, it lives in their lymph nodes. It lives in places that I can't reach with a needle without really causing some damage. So the other part is that we've been doing autopsy studies for HIV since the beginning of the epidemic, but the problem is that they were always done too late. So people would come in, they would pass away, they'd be put into a freezer, and then the autopsies would be done when it was convenient. And that really allowed HIV to escape. Those cells died. The virus was able to go into the ether just through the degradation and entropy process. So we never quite understood exactly what we needed to know to cure HIV by just doing previous autopsy studies. The next one is we decided to come up with this program called The Last Gift, based on my patient's desire. And we were looking for altruistic people with HIV who had a terminal illness. And they had all sorts of things, everything from heart disease to ALS, which is Lou Gehrig's disease, to cancer, to asthma, to all sorts of things. But it was at the end of their life. And then we were going to follow them while they had this terminal illness. We would take some blood, we would take some genital secretions, we would take some stool, anything that you could imagine in terms of things that we could look at in the lab. And then when they passed away, we would do a rapid autopsy. And what that means is that within 6 hours of death, we have somebody already dissected and put into little tubes. We have collected all the material that we can and then frozen it in a viable way so that we can collect as much information as possible throughout somebody's body. And this was pioneered originally in cancer research using new therapies. And we were just repurposing that for people with HIV. And it really did preserve the quality of proteins and virus, etc. And this is the age range. So you can see that we have quite a few older people, but if you also notice, we have quite a few people who are younger who participate. And what we've noticed is that the younger someone is who receives a terminal illness, the more likely they are to actually participate in the study. These are the comorbidities along the line, everything from heart disease to cancer to different types of cancer to bone marrow cancer, etc. This is the time that somebody could be in last gift, even though doctors say you only have 6 months to live. Oftentimes it's sooner, just a few months. But we have some people who've been out to 4 years within the study. It's been funded since 2017 by the NIH, and we just follow people the whole time. Now, one of the questions we get asked a lot is how do the participants feel about participating in the last gift? And this is actually our community advisory board. So we actually, our board and participants and next of kin and family and friends of previous participants. We invite them every year to meet together and to talk about the study and have a good time giving back the science to them, to tell them what the contribution was to the scientific world. So that's a nice picture. We just had ours the other day, by the way. It was very nice. So how do people feel? So the participants very much feel my death will not be in vain. We have all these testimonials. This is my last gift to science. One day I'm going to be able to make a difference, even if I'm not around to see it, etc. We also ask the next of kin, and they have the same perceptions, that they feel proud of their their loved one who gave this gift to us. And then we also interviewed the study staff and the researchers who do it and how much it is valuable to be able to say that I continue to hold this gift valuable for science. And I want to make as much meaning out of what they've given us into scientific discovery. Another interesting thing about how do people feel about it, so these are people who are at the end of their life. So now we're going to measure quality of life scores. And as you can imagine, someone who is dying don't usually have high quality of life. And as they approach death, they usually feel worse about their quality of life. It turns out that once somebody comes into our study, we actually see the quality of life scores go up. And quality of life scores going up means I have more quality of life. I feel better about what I'm doing. And when we ask them why you feel better, even though you are getting sicker, usually they're bedridden, having to go to inpatient hospice, etc., they always point to, I feel like I'm giving back. I have meaning in the last moments of my life in making a difference. It's not wasted anymore. So the other measure we did was called a Beck depression score. So oftentimes when people are dying, they get depressed. And you can clearly see that on the Beck depression score. And it turns out that the higher score is more likely to be depressed. So the other one, the higher score was better. Here, the lower score is better. And we saw exactly the same pattern, that people got less depressed the closer they were to death while they stayed in the study overall. Why? Because I felt like I was doing something for science. I had meaning towards the end of my life. And that always made us feel good. And then we saw a correlation, as you would expect, that quality of life and the Beck depression scores went together. The better somebody felt about their quality of life, the less depressed they were. And what it gave to us was that this wasn't just about altruism. This wasn't just that people wanted to give back because they didn't get anything out of it. They actually did get something out of it. They felt better about the last moments of their life in terms of the meaning that they were able to provide to society. Okay. This is LGO5. He wants us to use his name. We have quite a few of our participants who were very clear. They wanted their name attached to the study, and we are going to honor that request. And this is Max, and he was in the show business in Hollywood for a long time, and he definitely was a star in many ways. But what do we measure before somebody passes away? So we have a lot of what we call ante-mortem data, and this is, since we know who they are, we have medical records that go for a long time. We can get viral loads, we can get CD4 counts, we can get all sorts of therapy that somebody has taken along the way, and we can get the full history of somebody's medical. And some people get on therapy here. This is LG34. They start therapy, they stay suppressed for their whole life. And that's the magic of antiretroviral therapy at the moment. HIV no longer kills somebody if they can take the medicines, and if they have access to those medicines, they work. That was the reason my patient was able to go home and could avoid my lumbar punctures after a while, right? He lived to die of something else. Some people, when they come into the study, this is LG30, their viral load is all over the place because they stopped their medicines. They get to the end of their life and they're like, I'm tired of taking my medicine. I don't want to take it anymore. I don't see any point in it. And yeah, okay, stop your medicine. It turns out that was exactly what happened to my patient, my very first patient, who gave me this idea. Even before we had the study, he didn't want to take his medicines anymore. He stopped them. So then that gives us an opportunity to look where the virus goes when it rebounds. That's what it's called. So they're on therapy, it's suppressed, and then when it comes back, it's going to go populate all the different tissues throughout the body. So it gives us a new opportunity to look at that as well. Other things that we're very interested in are substances. So people take all sorts of things in their life. This one is tobacco. Some people take stimulants, sedative. We're doing our program in California. Lots of people smoke marijuana. We have everything down here. And also, when somebody's at the end of their life, they have a lot of pain and take a lot of opioids. And this is a lot of pain medications where we can actually march out when they're taking it, how they're taking it, etc. Because when they have the autopsy, we want to see how much of that gets into what tissue, because it doesn't get into the tissue all the same way, and it doesn't cause all the same effects in all those different tissues. So we're for the first time learning about what these different substances can affect different tissues. Okay. Then the person passes away, and we get a call, and it can happen any time of the day. It turns out that we have most of our deaths happen in December, and it happens in the middle of the night. The first thing we do is have our moment of silence, and this is what we read at the bottom for every one of our autopsies. And we get everything together and ready. At this time, this is me out without my hat, but we're putting stuff together. The body is off here to the side. You can't see them. And then here in those trays are the various organs. So somebody, what we call a diener, someone who's doing the autopsy, is taking the pieces of tissue as fast as they can, as fresh as they can, and we're putting them on ice. And then us in the middle here, that's me, are cutting up the tissues in very small pieces to get ready to freeze them, to put them in RNA later, basically different preserving ability to catch as much as we can about what's going on in those tissues at that time. Okay. At the end of the autopsy, we send those tissues all sorts of places. So here is Jim Dunn, somebody else who wants us to know his name. And he passed away last year, and this is at the LGBT Center, and they're giving us an award. That's Sarah Janella. I think I'm behind there. There I am. And giving us an award about the Last Gift program. And he was a longtime activist. That's his wife, Susan, by the way, in the HIV community. And he was very proud of being a participant in the study and was one of our biggest supporters. And he passed away in the study, and that's him. And we put him in a whole bunch of boxes, and we sent him all over the place. So, because we can't do everything at UCSD. There are techniques that are in Harvard and Hopkins and Gladstone virology that we just can't do at UCSD, but we want to make sure that Jim's gift is as powerful as possible to learn as much as possible. So we send them out that night. That was him on the loading dock that night. And if you want to know more about him, there's going to be a film, a documentary specifically about his journey with the Last Gift. It's going to be in a film festival in California, but it should be coming out soon for lots of places. So we're very excited about that. So back to what we're learning. So after the autopsy, we do a whole bunch of stuff with the different tissues. So I'm going to do a whole bunch of data dump. Hopefully it doesn't get too boring. I'm excited about it, so hopefully that'll be infectious. But one of the things as we look at is opioids and morphine. So a lot of people in the last parts of their life take morphine for their pain control. But it turns out that we learned a lot because morphine, even though they might take it, it doesn't go all the same places. So here's all the different tissues that we had checked. Everything: spleen, rectum, prostate, duodenum, frontal cortex of the brain, blood plasma, basal ganglia, adipose tissue, muscle tissue. And we see differences. And it turns out that the differences are consistent. It gets very high into spleen and liver, but it doesn't get very high into muscles. And there are morphine receptors that are different in different places, and that we're now learning is why some people might respond better to morphine or some respond better to tramadol, etc. So just even learning about how opioids affect the human body has been a big push now in the Last Gift program. And that doesn't have anything to do with HIV. We can also look at antiretroviral therapy and how it gets into all these different tissues. And this is the level of, here's the central nervous system, gastrointestinal tissue, genital tract, vascular system, etc. And you can see that, boy, antiretroviral therapy does not get into the brain very well. And we have seen this previously, that virus can sometimes escape from the brain. And when somebody's taking antiretroviral therapy, the virus can sometimes get out and cause damage in the brain. And we're going to talk a little bit more about that. But it's likely because the antiretroviral therapy just doesn't get there in some people high enough to control the infection. And then we can also look at inflammation. So we do a whole bunch of cytokines, and that's meant to just be overwhelming number of inflammatory markers that we measure. And this is all done by also a former postdoc of mine, Antoine Chalone, who's now a professor also in his own right. And this is one of his specialties, is to look at all the different tissues and all the different concentration that actually happens there. The other thing he does is measure HIV, and HIV comes in two forms. One form is called HIV DNA, and the other form is called HIV RNA. It's a retrovirus, so it has an RNA form, and then it turns into a DNA form, and then that DNA form, it goes into the human host DNA. And once it's in that DNA, it is hard to get out. That's the whole reason that HIV is hard to cure. So when somebody takes antiretroviral therapy, it knocks out the RNA component, but the DNA still survives. And this is going to be important for a later part in the talk. But that DNA is the reason why we have not been able to cure HIV. Like SARS-CoV-2, it was only an RNA type, so we were able to knock it out. But here, the DNA becomes our own DNA during that infection, and then it stays. But we can measure it, and we can measure it in a whole bunch of things, including the brain, the gut, the genital tract, the lung, etc., and we can see differences along the way. The other thing that we can do is sequence it. And we can sequence the virus in its RNA form, and we can sequence it in its DNA form. And when you do the sequences, HIV evolves. In fact, it evolves so well that every person that has HIV has a different genetic variant of DNA. In fact, it's so different that I can tell when somebody got HIV from one person to another person just by sequencing them. And I can see that there are some connections. It's just like knowing that somebody's mom and dad and brother. I can detect that with two HIV variants from two different people. And then we can use that to our advantage on the sequencing to see where HIV goes within the body, because it starts to evolve when it goes from the brain to the blood to the liver to etc. And then we can start to track it. And that's what Antoine did here, and was one of our first biggest studies to show that it was the blood. So the peripheral blood mononuclear cell, which is called PBMC, was giving rise to all the different parts of the body. Hippocampus, colon, jejunum, rectum. It was all mostly related to the blood. And this was when somebody was on therapy. So somebody had taken HIV therapy, there was no HIV RNA in their blood, it was only DNA, but the cell that was infected with that HIV was basically trafficked to the brain, trafficked to the colon, etc. Then we looked at somebody who had turned off their antiretroviral therapy, and it got messier. We could see the HIV virus going a whole bunch of different places, but it turned out it was still the blood. The blood was giving rise to all these different places. And it answered a big question for us, because there have been cures of HIV. Does anybody remember the Berlin patient? So the Berlin patient, a long time ago, got a bone marrow transplant that made his blood impervious to HIV infection. But that didn't stop all the virus in all his other places, the liver, the spleen, the brain, etc. But he was cured. We couldn't find the virus in his blood after the antiretroviral therapy was turned off because the blood was impervious. So this trafficking that happens when somebody turns off their therapy didn't happen in him because the blood highway was turned off. So it answered us a big question that if for an HIV cure, we really did need to focus on the blood. Okay, I want to talk a little bit about the brain. Actually, I'm going to talk a lot about the brain because we've always known that people with HIV, even when they're on therapy, have neurocognitive problems, a lot of them. Almost half of the people can have neurocognitive deficits. Things that you might not ever notice, but we can figure this out through testing. And we don't know why. So the first thing we did was measure HIV DNA in all the different parts of the brain. Basal ganglia, hippocampus, occipital, also the spinal cord, which I'll talk a lot about in a second, and this is the blood. This blood is always higher in HIV DNA, but we can detect it in every single tissue that we can measure within the central nervous system. Oh, real quickly. This is Mattia. He's one of our postdocs who just got a nice professorship in Italy, so he's leaving us, which is very sad, but all this work is what he did. And we presented it this year. And what we found was that there were higher transcriptional levels, and this means that HIV is changing from its DNA form to its RNA form. It is forming HIV RNA, and that is popping off viruses within these cells, and it's specifically doing it in the basal ganglia and the hippocampus. And these are areas that are very important for how we store and how we make memory, actually. And what's happening here is that somebody's on therapy, but the virus is popping off. And that popping off is causing inflammation. And we'll talk about that in a second. But that inflammation is causing damage in those areas, even though somebody is on antiretroviral therapy. The other thing that we noticed was that the spinal cord itself was a sanctuary, and this is very new. We didn't think that there was very much going on in the spinal cord, but it turns out when we sequence the virus, and this is a phylogenetic tree showing all the differences of different HIV variants that we look at different parts of the brain in the blood and the spinal cord. And what we saw was that there's always these little populations of viruses that are different than the other rest of the body. And we call this compartmentalization. And we saw this very specifically in the spinal cord. And what that told us is that the virus is going into the spinal cord and it's learning how to live there. It is evolving to its environment specifically, and that is different than all the other parts of the body. And then we can actually see that across multiple people. So like 25% of people will have different viruses between the spinal cord and the brain, while 37% of people will have differences between the spinal cord and the blood, and then between the brain and the blood, almost 3/4 of people, the viruses genetically look different. So the viruses are changing to adapt to those new environments within the same person. Well, that sounds horrible, and it is. But it also gives us a way to track the virus. So now we can start to figure out, we can use evolution against the virus by tracking it through the body. And just like we did in all the different things, we can do it even in the spinal cord and the brain. And what this shows here is the donor site, and here is the recipient site. And we can see that the spinal cord virus jumps over to the occipital lobe or the basal ganglia. But it also goes back to the spinal cord in different places. And then we can really look at it very closely, where we can see where the spinal cord gives it to the spinal cord. So if this is the donor and this is the recipient, this is the basal ganglia gives it. The spinal cord gives it more often than it gets from the basal ganglia. And then the hippocampus is exactly the opposite. And the occipital lobe turns out to just be a sink. The virus comes from the spinal cord into the occipital lobe, and the occipital lobe never gives it out. And this is all interesting, right? Except it's not just academic. If we're going to cure the virus, then we need to know what the virus is and where it's going in somebody to be able to cure it. And now we know that the spinal cord, just like the blood that I talked about before, is the highway for the brain. So if we're going to do a cure and we're going to get rid of HIV in the central nervous system, we can't ignore the spinal cord. Okay. But where in the brain is it happening? There's not many CD4 cells, which are the primary cell that holds HIV in the brain. It turns out that the immune cell for the brain is called microglia. And it is sort of this glue, small little glue cells, that's what microglia mean. And people have long thought that they are the reservoir for HIV, but nobody could prove it. Why? Because every time they took somebody's brain out and they tried to get the cells out, the brain was too old to be able to get live cells out. But all of a sudden, we had brain that was live enough to where we could pull out microglia, and that's exactly what we did. Well, we didn't do it, but we sent it to North Carolina, where Dr. Guchan and Dr. Yu Yang were able to pull out the microglia because they are experts in that. We were not the experts, so we sent those brains overnight to them. They pulled out the microglia, they grew it, in cell culture, and they gave it a little push called latency reversing agents, and bam, the virus came out of the microglia. So for the first time, we could see that the microglia was the reservoir for HIV in the central nervous system. And this is just to show that for every participant that we had, we could grow it out of the microglia, and we did not grow it out of the CD4 cells that were in the brain. There were just not enough. The microglia was the reservoir that actually grew out live virus. Okay. Now, the other question that we had is, well, does that actually mean anything? And this is Dr. Johannes Szecki, who's at UCSD, and he's also a single-cell RNA-seq expert. So what he really likes to look at is how well does HIV express within a single cell. So we sent him some brain and said, hey, I want you to look at the expression of HIV in these cells. And what he did, he first looked at HIV DNA, and he could look at HIV DNA in these cells that he mapped. So in a cine plot, basically, DNA was just randomly dispersed. It really didn't matter. HIV DNA was everywhere. But when he looked at RNA, so when the virus was popping off, changing from its DNA form to the RNA form, it localized in this little quadrant over here. And it turned out that this quadrant was the inflammatory quadrant. And you can imagine that the virus pops off, the cell recognizes, oh my gosh, I'm infected, I need to do something, so I become inflamed. And oftentimes those cells die. And if you're thinking about that in the brain and you're having brain cells die, it's not a good idea. And that could explain why we're seeing more of these neurologic and neurocognitive effects in somebody who has HIV, who's on therapy, but they're still having virus that's popping off, causing inflammation in the brain. Okay. Going to switch gears. So now we're going to talk about the microbiome. So we have whole body, so why don't we just do a whole bunch of stuff? And this is another thing that Matteo has been looking at. And we just sequenced the crap, pun intended, out of every single part of the GI tract. So everything from the duodenum to the rectum, we took a little bit of poop that was in all those different places. We did deep sequencing to figure out what was the bacterial species that were there. And it turns out that they're different. So in the duodenum, you can see what we call Enterobacteriaceae more often, and those species become more diverse as we get further down the GI tract. That was new. That was new to the field, to see that overall our GI tract is not just one long thing of the same kind of bacteria. There are environmental niches that are different for each part of our GI tract. So that was pretty cool. Then the next thing we did was to do HIV DNA, that's what this is, in all the different parts of the GI tract. And we did HIV RNA in all the different parts of the GI tract. So you can see like the duodenum has a lot of HIV RNA, even when somebody's on therapy. It didn't have that much DNA, but it had a ton of RNA, while the jejunum had a ton of DNA, but no RNA. We're still trying to figure that out, by the way. Why does the jejunum have a ton of DNA, but it doesn't allow HIV to pop off? That might be something to pursue, while the duodenum has the opposite. Not as much DNA, but it pops off all the time. So one of our first thoughts that perhaps it was about the microbiome. And it turns out that the more species of bacteria that we saw in a particular place, like say the small bowel, so the duodenum, the jejunum, and the ileum, we saw more transcriptional activity. So the virus was popping off more, the more species that we saw there. So diversity, you could think of as bad for HIV. It caused more HIV RNA to pop off. And it actually turned out to increase inflammation as well, which actually could cause colitis, et cetera, in these people. But the opposite was true for the large bowel. So the more species you had in the large bowel, actually the HIV RNA was less. So what you want is to have less species in the jejunum and the small bowel and more species in the large bowel, the colon. And it turns out that's exactly what IBD researchers have found. So people who have irritable bowel disease, Crohn's disease, etc., people who have colitis have very similar bad patterns. If you have a lot of diversity in your small bowel, bad. If you have small amount of diversity in your large bowel, bad. So it's very interesting to see very similar patterns on the HIV side. The next thing we did with this is the group with Gladstone. They're experts at figuring out what cells do. So this is a complicated slide, but basically we were able to take tissue from a whole bunch of different places, lymph nodes, spleen, liver, and they had this program called CyTOF where they were able to put different participants into their CyTOF and they break it up into different things. Here's CD4 cells, CD8 cells, myeloid cells, B cells, etc. And then they break them up into different little pieces. They could pull them out and see what each one of those little cell groups do. And what they were interested in is how do those cells respond endogenously to viruses? And do they all do it the same way? And it turned out they didn't. So when they did RNA-seq on these different groups of cells, they found that certain viral sensors were different in the lymph nodes versus in the mucosal cells versus in other places. And what that means is when HIV is in a CD4 cell in the mucosa, it's going to behave differently than a CD4 cell that's in a lymph node or in a liver cell. And that tells us a lot about how, if we're going to eradicate HIV from the liver using its own antiviral mechanisms like interferon, etc., we're just going to have to do them differently in each different cell type. So a hard problem that we already started off with now has gotten even harder. But at least we know that that's the universe which we're looking at. Another big one is broadly neutralizing antibody. So I don't know if you remember, but during COVID, we used these things called monoclonal antibodies. It was used a lot in my trial, and it's basically taking somebody's antibody, growing it up in the lab, and then giving it as a treatment to cure COVID. And it worked great. Is before we had other drugs like Paxlovid and Molnupiravir, but you had to get an infusion, etc. Now people are using that for HIV because we can get HIV antibodies that can knock down the virus. And they're being used right now in experiments and clinical trials for therapy. But the virus, as I said before, evolves like crazy. And it evolves away from antibodies pretty crazy fast. And it turns out that it oftentimes evolves away from antibodies differently in the brain. And if you remember what I told you, it loves to evolve differently in the central nervous system. And in fact, it's evolving differently away from different broad spectrum antibodies. That's what these are. So let's say this person were to get this particular antibody, it would get rid of all the virus we found in the blood, the gut, the liver, the lymph nodes, but the one in the brain would continue to grow. because it was naturally resistant to that antibody. So now we know when we're using these therapies for people and we don't know what's happening in their brain, we might need to check on that while they're getting that therapy, because we might just be missing a whole bunch of virus that's growing there. So another thing that we're able to do is to track our immune environments over time. So here's a participant who we can see a whole bunch of different T-cell receptor clonotypes that happen over time, and this is what this means. But these are tracking to various infections, let's say cytomegalovirus, herpes simplex, a whole bunch of EBV, a whole bunch of viruses that we all have that are being targeted. And now we can map those to different tissues because they don't all stay in the same place at the same time. So we can see them differently in the PBMC versus in the occipital cortex versus in the kidney, but they also move. So the ones in the blood might move over down to the ileum, or it might move over to the heart. So we can start to track the immune response in the body over time based on their T-cell repertoire. And we can do the same thing with viral migration, which I showed early on, that we can see the virus move between the blood to the prostate, to the rectum, etc. And then we can overlap the two. And that's exactly what we're seeing, is that the immune response is following the virus in different places. We can see this migration happening, both in the immune response, but the other thing that might be happening is that the very cells that are supposed to control the virus, the T cells, are immune cells themselves. And they're also moving to different places. So here we have a T cell, it's infected, and it just leaves the blood and goes to the brain. And now we can track both its viral movement, but also its immune movement. Okay. So now I'm going to tell a story, but this is Tony and Blake. This is LG01. They wanted us to use their name. And so they're our very, very first participant in the last gift. And if you want to see their story, it is on our website, lastgift.ucsd.edu, and there's an interview with them, and it's very endearing. But that's LG01. And he was dying of ALS, but I'm going to tell a story now of cancer. And this is a patient, participant that we had who was 74 years old. He didn't smoke, he didn't drink alcohol, he got diagnosed in 2000, and he developed lung cancer. And when he had lung cancer, he had a bunch of chemo over time, but it just wasn't going to work because it had spread to lymph nodes, to the spleen. So he entered into the Last Gift project, and he passed away in February 2022. Okay, this is going to be his story. Now, the interesting thing about cancer is that people with HIV get more cancer than other people. Even people who have HIV, who are on therapy, who are doing great on their therapy, get more cancer than other people. And it's not just AIDS-related cancers. It's not just Kaposi's sarcoma or primary lymphoma. It is all types of cancer, breast cancer, lung cancer, etc. And we were wondering, why would they, with the seemingly normal immune system, get more cancer? And that led us to really think hard about what's called the tumor microenvironment, and that's what this is supposed to represent. But in there, we have a blood vessel, we have some virus running around, we have some bacteria, we have some immune cells, we have some tumor cells. Tumors develop, and then the immune cells are supposed to go in there and get rid of the tumor. In fact, every day we're developing tumor cells in our body, and our immune system does a great job at going in there and killing them until it doesn't. And that's when cancer takes hold, and it's usually in this microenvironment. So we went deep into that microenvironment. And in this participant, we looked at his blood, we looked at his spleen, we looked at his brain, and we looked at parts of his brain that had tumor, and we looked at parts of his brain that didn't have tumor. And what we found was that those with the tumor versus those without the tumor, those with the tumor had a lot more HIV in it. That's weird. Tumors don't get infected with HIV. Only the CD4 cells that are in there are infected with HIV. But every time that we looked, in all the places that we looked, we saw more HIV DNA in the tumor versus the normal tissue that was sitting right next door. Okay, so that was weird. So then we said, why? What is going on here? So we went back to think about what happens with HIV. So HIV in its RNA form changes to a DNA form, and it inserts itself into the genes of the person. And it does this whatever genes are open. So at any one time, we have all these genes that are open, and HIV, if it sees an open spot, will plug itself in there. But when it plugs itself in there, what does it do to the gene that it just plugged into? It breaks it. And when it breaks that gene, that cell no longer can use that gene. It turns out that a lot of those genes that it plugs into, we need, and especially for CD4 cells, it makes them functional. And what we saw was that in those tumor microenvironments, that's the TME, there were a lot of these genes called P10. There's also this one down here that's in the green, that are important for the T cell to work. So basically what we had was a T cell that didn't work very well. It was lazy. It didn't do its job. It couldn't clear out the cancer. It might be attracted to the cancer, so it runs into the cancer, into the burning building to clean it out, but it's lazy, so it just sits there, and it takes up space so that good T cells couldn't come in to do their job. So now we have, this is our working hypothesis, is that the T cells that are in these tumor microenvironments are just not doing their job. And when we looked at it through expression, we could see that in normal tissue, that's right next door, we could see lots of expression of EGFR and P10. That is the normal production of genes that would help clear out a tumor. But within the tumor, we saw none or very little of those two genes expressed because HIV had inserted itself into the T cells to keep it from being produced, which we think is our hypothesis why it was able to cause cancer in this individual. Okay. We get asked a lot. I'm going to turn back to the human side of it. We get asked a lot about our ethics. We have a bioethicist. This is Karina Dubé. And she's been so prolific about our study and doing surveys and really talking about the bioethics of doing these studies at research at the end of life about how these participants, if they can decide to go to the grocery store or drive themselves or do their own bank accounts, how come they're so vulnerable enough that they can't do research? And this push is now, we've had many discussions with the FDA in terms of using this as a model for research at the end of life. So it's now pretty well established that these participants are not vulnerable. And this is our community advisory board, Jeff Taylor and Andy Cates, who really have led the way of talking about how people at the end of their life are not so vulnerable that they can't keep making decisions for themselves, even when that is research. And one of our big components here is that we do have a community advisory board where we talk about these things and every participant a lot and how things might change. So sometimes people might be vulnerable closer to the end of their life and we pull back or we figure out how to keep honoring their wishes at the end of their life. But this is really all based on our community advisory board. Okay. So let's go back to some crazy questions. I showed you a whole bunch of data. This is the big push that gets me excited that hopefully we can figure out how to cure HIV someday. But we also have other things that we could do. In that first study that I showed at the beginning where I asked people how many days, how many months, how many weeks that they'd be willing to give up, I also asked them would they be willing to me to infect them at the end of their life. Would you be willing to let me give you strep throat, HIV, hepatitis C, malaria at the end of your life for research? I asked that question. And the reason I did that is kind of nuts, because I could learn right away if an HIV vaccine worked, or strep throat worked, or hepatitis C therapy or a vaccine worked right away. I wouldn't have to do 1,000 people study. I could just have 12, and I would know right away whether something worked. And what was interesting is that, yeah, a third of people said I could give them strep throat. And I just want to let you know, I would not want strep throat. That's the one on here that I would not want, because it actually hurts. The other ones, you probably wouldn't notice. That would really be a game changer in how we did it. But when we went back and asked, that was the one they were most familiar with. They had most often had strep throat, so they knew what they were getting into. So yeah, they were willing to do it. Then we asked them, years later, a whole bunch of different group, whether or not they would be interested in a first-in-human study, whether or not they'd be interested in therapeutic vaccines, transplantation, cellular therapy, genetic manipulation of cells, and over half, basically, would be willing to undergo various interventions at the end of their life if it would help somebody else. Not them, they know they're going to die, but they're willing to do it for science. So no matter how you're feeling about the human race these days, this should give you hope. Here's some of our considered interventions, everything from neutralizing antibody to CAR T cells to latency reactivation agents, drug penetration, imaging, vaccines. Do you have ideas? You want to do collaborations with us? We're in the discussion with the FDA for a lot of these already. Think of how many animal studies have failed because they're just not human and they can't consent. Humans can consent and they're human. So their immune system would be so much better at figuring this out. So this is one of our last gift community meetings where we made what they felt about our study, and these are the big words that how often they were used: engagement, gift, participation. And this is my team. So thank you.
So we have time for questions.
Hi, I'm Federica Darima, and I'm a member. And thank you very much for this work that you're doing and for presenting it to us. I have many questions, but Larry only allows us to ask one.
Well, you can ask
So
two.
what is it in the spinal cord that enhances the rate of mutations and the diversity of mutations?
Good question. And I will start off by saying I don't know. My hypothesis is that there are different cell types that cause the virus to evolve to infect those cell types. So the microglia in the spinal cord seem to be different than the ones in the brain, and definitely different than the CD4 cells that are circulating in the blood. So the virus needs different entry and other ways to grow there, and that's causing the evolution.
And you said we can ask one more question. I mean, this is the study that you do for HIV, but one can envision that can be done for many other diseases. You talked about cancer in relation to HIV, but it could be also studies for cancer per se, right?
Yes.
Related to HIV. Are there any? Do you have any plans for that, or are there any others that are looking?
Yes. Thank you for that question.
Thank you, Larry.
So we've submitted cancer grants and diabetes grants and heart failure grants and rheumatology grants. And all of this is it's a research model that we could use for lots of different diseases. Oh, and we have a grant for ALS.
Chances to be funded given the funding environment today?
No, let's skip
This is my
that
third
one.
question. So
No, that's speculation.
We're working on it.
Red microphone.
Ciprian Ivanov, member. Does radiation therapy show more success relative to the HIV positive population than other treatments? And if so, does that have various effects on the HIV condition as well?
Good question. So radiation, so for cancer therapy, radiation is used a lot, as is chemotherapy, and they both are horrible for people with HIV. People with HIV who develop cancer are often resistant to both of those. And I don't know of any differences in terms of HIV direct effects for radiation, but I do know that radiation just doesn't work very well for people with HIV.
Blue microphone.
Hi, Umberto Caldelas member. Thank you again for the talk. Can you go into a little bit more detail on why there's a fundamental difference between freezing someone's body after they die and then doing the autopsy versus cutting them up and then freezing them? Is it because you can tailor how you freeze individual tissues? In my mind, it's just genetic material, but I'm an engineer, right? So can you kind of elaborate on why the specific method that you chose works?
Yeah, good question. So, and I should explain more, and thank you for that. So when somebody's frozen, just in general, it's done like in a refrigerator at -4 degrees or 4 degrees. When we freeze it, it's -150. We're using liquid nitrogen, and we have very special techniques to keep the cells still alive when we're doing that freezing. So there's a controlled rate freeze, there's special formula that we do, and that has to be such small bits of tissue that all that sinks in there to be able to freeze it in the right way to keep it still alive.
Red microphone.
Hi, I'm Caitlin Lucy, member, and thank you first and foremost for this wonderful talk and all this wonderful work as well. As it pertains to the microbiome gut studies and the different signatures of RNA and DNA in different segments of the gut, I'm curious if you've looked yet at metabolomics and what these microbes might be producing that has those implications.
I love that question. So yes, one of my colleagues at San Diego State, she sent me a whole slide deck yesterday. We've been doing metabolomics now for about a year, and we do see differences in the different parts of the GI tract, but also in the heart, in the different parts of the fat tissue as well. But in the gut specifically, the metabolomics does the inflammatory rates are related, and the metabolomics do look to be correlated with the amount of species of bacteria that are in the gut.
Thank you.
Is the blue microphone?
I'm Sina Sabit, and I'm not a member. Question about whether CRISPR has been looked at to take out this HIV DNA out of the genome.
Yeah, it's a good question. There are groups that are looking at CRISPR, but other molecular type scissors to take out HIV genome. It is all about the delivery to be able to get the little scissors in the right place to be able to cut out the HIV. And perhaps you don't have to cut out the whole thing. You just need to cut out a piece of it so that it doesn't grow anymore. And people are definitely looking at that, and we have been in discussion with the FDA to do it specifically in this cohort. Because what we're interested in is, where does it go, right? So in somebody who's alive, you will never know if it went to the brain or not, because you're not going to do a brain biopsy. But in this group, we could give the CRISPR or whatever molecular scissor it is, and then when they pass away, when we do our autopsy, we can go and see, did it go to the right place in the spinal cord, the brain, the liver, etc., and how well did it penetrate? Because we already know from our opioid studies and our therapy studies, the distribution is just not going to be the same.
Red microphone.
Hi, Chancellor Groats, guest. And I was curious about how extensive the research has been between the duodenum and the jejunum, and why you think the jejunum is so inhospitable to HIV.
Yeah, I really don't know. So what we see is that the jejunum just doesn't let HIV pop off, versus the duodenum is constantly popping off. They are right next to each other. So we're reaching out to gastrointestinal experts. I'm not a GI expert, and trying to get them to look specifically at those tissues, because that might be a really important thing. If we can figure out why the jejunum does not pop off, and we can apply that to other parts in the body, that might be our cure right there. We don't have to necessarily eradicate HIV. We just need to keep it from growing. So there's one group called kick and kill, where you kick the virus and then you kill it. Another group is called lock and stop. You just lock it down and don't let it grow. And if you could do that, you would also have a cure. So that's one of the areas that we're targeting.
Sorry, isn't that something that CAR T cells do? With just, essentially marking something as itself so that it doesn't attack itself, essentially. So could that be applied in that area?
So we are definitely looking at CAR T. We have a research program specifically around CAR T, not in this last gift program, but we are looking at that. What I will tell you is that CAR Ts are blood. So while they will lock down the blood, there's other places that they just don't get to. And maybe that's all we need, but I think we're going to have to figure out something that's able to get into these deeper reservoirs on cellular therapy as well.
Got it. Thank you.
Blue microphone. Oh, Lloyd, please.
Yeah, I have a question. Lloyd Mitchell, I'm a member. I have a question about in the brain. So what you said is the HIV seems to primarily be in the microglia. Is there much of a reservoir in the neurons themselves? And I have a follow-up question, which would be, what would happen if you knock off the microglia? Would the brain, grow new microglia and, get better?
So good question. So no, it's not in the neurons. So it just doesn't get in. It might be in the astroglia, which we're still trying to figure out. In the microglia, it's only 1 in a million cells that actually have HIV. So we can knock off 1 in a million and probably not be too much in terms of damage. And yes, microglia can repopulate themselves to some extent.
Have some questions from our listeners online.
Thank you very much for your talk. We have one question from Dave Rabinowitz, a member from Corvallis, Oregon, who asks, are you listed on the NIH's clinical studies website, and where else can people look to learn more about and contribute to the last gift?
Wow, I love it. So yes, we're on the NIH website. We're also have the website lastgift.ucsd.edu. Please take a look, and you can just Google Last Gift, and we're up there almost immediately. So thank you.
Thank you.
Is another microphone out there somewhere? I'll ask a question while they're doing that. Is HIV integration promiscuous? Or has it got affinities for certain genes and not others?
So the first studies, that's a good question. So the question is, does HIV integrate willy-nilly, promiscuous, or has a predilection for places? So at first we thought that it integrated into what's called oncogenes. Now oncogenes is a weird name because they're related to cancer, but they're also very open. And what that means is the chromatin is open, and that seems to be the biggest predilection for HIV to come in. So if the genes are open, then that's what hits first.
So open chromatin usually denotes active genes.
Yes.
So some of the differences that you see might be which genes are active in which particular cells.
That's right.
Now, does adjacent... Oh, I'm sorry, I'm asking too many questions. Let's go to the red microphone.
Ciprian Ivanov, member, is there variability in the P10 cells that become defective?
We don't think so. Good question. So it looks to be all those cells that had P10 integrated into them all came from the same cell at the beginning. So there was one cell that HIV integrated into P10, and that one clonally expanded all through the body. And that is actually the biggest one of the biggest issues with HIV reservoir is that during the first stages of HIV is integrating in all these cells all these genes that are open. A lot of times that kills the cell because it interrupted a gene that's open, that's active, that needs it to live. But there are some genes that cause that cell to then be alive but just not work. And P10 happens to be one of those that it does. And that gives that cell a survival advantage. It clonally expands, it does not die.
Blue microphone.
David Rosen, member. This last gift program, do you also take contributions from people who are dying of other terminal diseases? Is this program only for HIV and related, or is it for terminal diseases in general?
Thank you for that question. We got a supplement this past year to include what we're calling controls, but people without HIV who are dying of other diseases. So now we have a whole control group, and then we're working on trying to get more funding on the diabetes side, especially around GLP-1 inhibitors for this group who don't have HIV to tackle other diseases. So yes.
Any other questions? Maria. Red
Maria
microphone.
Zemankova, a member, and I'm going to ask a very naive question. Are all people equally prone to getting HIV if they come into contact with HIV? If not, can we learn something from it?
Yes, we can totally learn something from it. So people are not all the same. So in 1996, they discovered that CCR5, this protein that is normally on T cells, was needed for HIV to get into that T cell. And there was a lot of people who have a mutation in CCR5 that does not allow HIV to get into that cell. And there's a great story about how it's survivors of the bubonic plague, if you want to know. And it's very fascinating. But from there, they made a drug called Maraviroc to knock out CCR5 in us who do have CCR5 that's normal. And that is an HIV drug that we still use today. So we can learn a lot from things that don't happen.
Other questions? Hearing no questions, we will thank our speaker. We have a few gifts for you, which are actually in the back of the room, but just so the audience knows what we're giving you. A framed copy of the announcement of your talk, a rosette, a PSW with 3 stripes on the side, symbolic of the unit for inductance, which was studied by the founder, Joseph Henry, and a copy of Volume 17, signed on behalf of all the members. You'll find 3 histories of PSW in there, as well as other interesting tidbits that I'm sure you'll enjoy reading on the flight back to your Christmas celebration. So thank you very much.
Thank you.
Before we go, we do have a few closing announcements. The next talk will be the 2528th lecture on January 9th of the new year 2026, for which we all have high hopes, I'm sure. The speaker will be Bernard Carlson of the University of Virginia and the University of Galway. He'll be speaking a bit on the life of Nikola Tesla and on the rivalry in the development of wireless communication systems between Tesla and Marconi. Bernard is the author of the definitive biography of Tesla, and it's sure to be a lively lecture informative of how transformative inventions like radio communications actually come about and the personalities that drive them. Other upcoming lectures are listed on this slide. The annual President's Lecture will be on January 23rd at the 2529th meeting. The speaker will be Gordon Guyat of McMaster University. He'll be speaking on GRADE, Rating Evidence Quality for Healthcare Practice. Gordon's the father of what now goes by the name evidence-based medicine, and a strong proponent of critical interpretation and cautious application of empirical studies in clinical practice. We will be updating the web page to reflect the addition of speakers as they come in, and I have their information. So please check there often to keep up with our schedule. About 10 days in advance of all events, PSW sends all members invitations to the pre-lecture reception and dinner with the speaker, and notices of the lecture itself to everyone on the PSW friends list. Feel free to join PSW and sign up for lecture notices via the website. You can link to the PSW calendar there as well to automatically sync to your PIM. The website is www.pswscience.org. Let's thank tonight's crew for producing this meeting, streaming and recording the program, and editing and posting the video to the PSW website and the PSW YouTube and Vimeo channels. Thank you. And with that, I will entertain a motion to adjourn the 2527th meeting to the social hour. Thank you, Bob. Do I have a second? All members in favor? All members opposed? The meeting is unanimously adjourned to the social hour.